When you look at a plant, maybe it's that potted fern sitting on your desk right now, or just the grass out in your yard, you probably just see passive greenery.
Right, just background scenery.
Exactly. It just sits there, soaking up some sun, drinking up some water. But today, we are going to completely shatter that illusion for you.
It is definitely an illusion.
It really is. Because hidden inside that seemingly quiet fern is, well, it's an architectural blueprint and an apothecary's journal that human engineers and chemists are literally still struggling to replicate.
We're barely scratching the surface of what they do.
Yeah, and for this deep dive, we are pulling from a really comprehensive scientific source on plant structure, function, and biodiversity. Our mission today is to decode the hidden engineering and the chemical genius of plants.
To extract those mind-blowing design features.
Right, and to see how humanity relies on them for sheer survival. I mean, from the microscopic structures that hold up entire forests, all the way to the modern clinical trials that save our lives.
Yeah.
Okay, let's unpack this. Because to really understand the macro-scale engineering of a forest, we actually have to start incredibly small.
Down at the molecular building blocks,
Yeah.
which I know sounds almost too basic, but a really profound shift in perspective happens when you see how a plant manipulates simple sugars.
There's this tiny, tiny chemical difference in plant biology that has just massive world-altering implications. Because both starch, which is how plants store their energy, and cellulose, which is what they use to build their structures, are made of glucose.
Right. They're both just chains of sugar.
Yeah, but starch uses what's called alpha-glucose, and cellulose uses beta-glucose. And it's literally just the placement of a few atoms on the side chains.
It's so subtle. In alpha-glucose, the hydroxyl groups, so that's an oxygen and a hydrogen atom bound together, they're oriented facing downwards. But in beta-glucose, one of those groups points upward.
Just one group.
Yeah, just one. But that minuscule structural variation causes a massive dilemma when the plant tries to link them together into a chain. They don't just, snap together easily like the alpha-glucose does.
Because the connecting points just don't line up anymore.
Precisely.
Yeah.
Because of how that hydroxyl group protrudes in beta-glucose, every alternating molecule actually has to be turned completely upside down. It has to be inverted so the bonding can even take place.
Wow.
Yeah, they form what chemists call 1-4 glycosidic bonds. When alpha-glucose links up to make starch, the molecules naturally curve, forming these compact coiling springs.
Which is perfect for dense energy storage, right?
Exactly. But because cellulose requires every second molecule to be inverted, those opposing forces cancel out any curving. So cellulose forms incredibly long, rigidly straight chains. And what's fascinating here is what happens next.
Okay.
Because you have these long, straight chains of inverted molecules running perfectly parallel to each other, hydrogen bonds can form horizontally between them.
So they link across.
Yeah. The partially positive hydrogen atoms on one chain lock onto the partially negative oxygen atoms of the neighboring chain. It's a process called cross-linking.
So you've got thousands of these sugar chains cross-linking to form a single microfibril, which then groups together into a fiber. So starch is like a tightly coiled spring of easily digestible energy. But cellulose, because of that inversion and the cross-linking, is like weaving unyielding steel cables.
It really is a masterclass in tensile strength built entirely out of sugar.
Which, naturally makes you wonder, if cellulose is technically just sugar cables, why can't we just eat grass for energy? Like, why isn't a lawn clipping just a salad that powers our brains?
I mean, that would solve a lot of problems, right? But it comes down to chemical locks and keys.
Okay.
Most animals, humans included, we simply do not possess the specific digestive enzymes needed to break those inverted 1-4 glycosidic bonds in beta-glucose.
We just don't have the key.
Right. We have the enzyme keys to unlock the alpha-glucose in starch all day long, bring on the potatoes. But cellulose, the locks are upside down. We just process it as roughage, dietary fiber. It basically squeaks through our digestive tract completely intact.
But wait, cows eat grass? Termites eat wood? They are surviving on those upside-down logs.
They are, but they're essentially cheating the system.
Cheating how?
Well, ruminant animals and insects like termites, they have specialized bacteria or protozoa living inside their guts. It's those microorganisms that actually manufacture the specific cellulose digesting enzymes.
Oh, wow.
Yeah, the cow is just providing a warm housing facility and delivering the raw material while the bacteria do the actual chemical unlocking to release the sugar cables.
That dynamic is wild. So the cow isn't even digesting the grass. The cow's microscopic passengers are.
Exactly.
So the plant has manufactured these molecular steel cables out of cellulose. How does it take those cables and actually construct its physical body? Like if we look at the plant as a growing skyscraper, how does it build the floors and the walls?
Well, it requires a highly organized, layered construction process. When a plant cell divides to form new cells, the very first layer it lays down is called the middle lamella. Think of it as the skyscraper's foundational biological mortar.
Okay.
And it's mostly made of pectin.
Wait, pectin? Like the exact same polysaccharide compound used to make fruit jam set and thicken in the kitchen.
The exact same stuff. It acts like an organic glue. It combines with positive calcium ions to make calcium pictate, which tightly binds the cell walls of adjacent plant cells together.
So it glues the bricks together.
Right, and once that mortar is set, the cells build their primary cell wall on top of it. At this early stage, the cellulose microfibrils, or steel cables, are all laid down in a similar parallel direction. This makes the wall strong but flexible, so it allows the cell to stretch as the plant grows.
But I mean, a skyscraper can't be flexible forever, or a tree would just flop over under its own weight once it reached a certain height.
Exactly. So the plant reinforces the structure. A secondary cell wall is built up inside the primary one. Here, the cellulose cables are laid densely at multiple crisscrossing angles to one another, creating a really rigid composite material.
Layering it up.
Yeah, and in woody plants, they pump in a complex polymer called lignin. Lignin basically impregnates the cellulose matrix, and it hardens like concrete. It makes the cell exceptionally rigid and totally impermeable to water.
Wow.
That combination is what we recognize as solid wood.
Wait, hold on. If the plant cells are systematically encasing themselves in these rigid, heavily lignified, waterproof concrete vaults, how are they not just starving to death in solitary confinement? Like they've completely walled themselves off from the rest of the organism.
It's a great question, but the plant actually anticipates the isolation problem during construction. It leaves microscopic escape hatches called plasmodesmata.
Buddhismata.
Yeah. They are incredibly thin threads of living cytoplasm that bridge the gap between adjacent cell walls. It's the skyscraper's internal wiring and ductwork, essentially.
Oh, okay, see.
These threads pass right through the newly formed walls, allowing signaling substances, water, and nutrients to pass directly from one enclosed cell to another. This creates a continuous, living, interconnected network throughout the entire plant, which is known as the simplest.
That is brilliant. So the plant isn't just a stack of isolated wooden bricks. It's one massive connected organism sharing resources inside this rigid scaffolding.
Precisely.
And that interconnected ductwork, it scales up brilliantly into the plant's main plumbing system found in the stems. I mean, the architectural blueprint calls for two entirely different transport tissues. You've got the xylem and the phloem.
The plant's internal superhighways.
Yeah. And the xylem is designed to carry water and dissolve minerals from the roots all the way up to the leaves. But to do this efficiently, the xylem cells actually undergo programmed cell death, right?
They do, yeah.
As they grow, they lay down that heavy lignin concrete in their walls. They become totally waterproof, and then their living contents just break down completely. The end walls between the stacked cells dissolve, leaving behind these long, continuous hollow tubes. It's literally dead biological plumbing.
It is, and those dead, rigid tubes pull double duty. They transport the water, but because of all that lignin, they also provide immense structural support to hold the plant upright against gravity. But the phloem, on the other hand, uses a completely different strategy.
Right, because the phloem carries the sugars made during photosynthesis from the leaves to wherever they are needed, so up to new buds or down to the roots. And because this is an active process called translocation that requires energy, the phloem tissue has to stay alive.
Right, they can't be dead tubes.
Exactly. The cells don't hollow out and die. Instead, they remain living, but they lose most of their normal internal organelles. And the walls between them become perforated like a sieve, which allows the sugary sap to flow through.
And since those sieve tube cells have sacrificed their own internal machinery just to make room for the sap, they have to be kept on life support by adjacent companion cells.
Companion cells?
Yeah, these companion cells are densely packed with mitochondria to supply the ATP energy needed for the active transport of those sugars. It is just an incredibly elegant division of labor.
It really is. But, I mean, a skyscraper's plumbing system, even one with dedicated life support cells, it needs pressure to move fluids against gravity. It needs a pump.
And this is where plant hydraulics rely on forces that are frankly astonishing, because non-woody plants, they don't have that thick lignin concrete to hold them up. They rely entirely on turgor pressure.
Okay, right. Because inside a plant cell, there is a large permanent vacuole. It's this fluid-filled space surrounded by a specialized membrane called the tonoplast. And the tonoplast meticulously controls what enters and exits.
Right. And the vacuole is filled with cell sap, which is a very concentrated solution of sugars, salts, and other substances. Because of that high internal concentration, the rules of osmosis dictate that water naturally wants to rush across the tonoplast to dilute it.
So as that water rushes in, the vacuole swells up and it presses the cytoplasm incredibly hard against the inside of the cell wall. The pressure that builds up acts like an industrial hydraulic pump. I mean, the pressure inside a tiny leaf cell can reach 1,500 kilopascals.
It's massive. To put that in perspective for you, human blood pressure, so the force of your heart actively, violently pumping blood through your arteries, is only about 16 kilopascals.
Wow.
And a standard car tire is inflated to about 200 kilopascals.
So the pressure keeping a delicate little leaf stiff is over 7 times greater than a car tire, and nearly 100 times greater than the pressure pulsing through our own veins.
Exactly.
That is insane. And if the plant loses water, that internal pressure drops, the cells become flaccid, and the whole structure just wilts.
Yeah, and that constant upward rush of water, which is driven by evaporation from the leaves, pulls essential minerals out of the soil and distributes them through the xylem.
So the plant is basically mining the dirt to fuel its chemistry. It pulls up nitrates to synthesize amino acids, without which it couldn't build proteins. It pulls up calcium to mix into that pectin mortar in the middle lamella. And it pulls up magnesium, which sits at the vital core of the chlorophyll molecule, making the capture of sunlight even possible.
Right. Without magnesium, the plant just can't trap light energy. The older leaves will turn yellow as the plant literally cannibalizes its own chlorophyll to survive, and eventually it just starves.
Well, here's where it gets really interesting. Because plants don't just meticulously pull up the specific nutrients they need for survival. Their root systems will vacuum up almost anything dissolved in the soil water. And scientists actually realized this hydraulic mining system could be exploited for something called phytoremediation.
Yes, this is a fascinating application. In the early 1970s, researchers who are analyzing flora in New Caledonia discovered a tree that produced a thick, bright blue sap. And when they ran the chemical diagnostics on it, they found that the sap contained 26% nickel in its dry mass.
Wait, 26%? The tree is basically bleeding liquid metal.
Pretty much. It's what we call a hyperaccumulator. Its internal chemistry allows it to pull massive amounts of heavy metals out of the soil without poisoning itself. And today, we actively harness this mechanism. In places like California, farmers will purposefully cultivate specific hyperaccumulating plants, like Streptanthus polygaloides, on soil that naturally contains high traces of nickel.
So instead of bringing in massive diesel excavators and destroying the landscape to sift dirt for metal, they just let the plants grow. The plants naturally suck the nickel up into their leaves and stems. Then the farmers just harvest the crop, burn it, use the heat to generate electricity, and sell the leftover ash, which is then smelted down to extract pure nickel. Farming weeds for metal literally pays better in some areas than growing actual food crops.
It's incredible, and the applications go far beyond profit, too. We use phytoremediation as an environmental cleanup tool. We can deploy specific plant species as biological vacuums on highly toxic land.
Wow.
Yeah, they pull dangerous heavy metals like thallium and lay directly out of the polluted dirt, locking it in their tissues. Land that was hopelessly contaminated from industrial waste can be completely restored simply by letting plant hydraulics do what they naturally do.
I mean, if we were already mining the plants for metal, it makes total sense that humanity would look at the rest of this incredible structural engineering, the cellulose steel cables, the lignified wood, those thick-walled sclerenchyma cells that give stalks their rigid snap, and figure out how to harvest the architecture itself.
Absolutely. Harvesting plant architecture is really the foundation of human civilization. We've always extracted the longest, toughest fibers, specifically from the sclerenchyma and xylem tissue, to manufacture ropes, paper, and textiles.
Things like jute, flax, and hemp. And cotton, where the fibers packed around the seeds are almost pure cellulose. We've been spinning ready-to-use plant architecture into thread for millennia.
Millennia, yes.
But in the 20th century, we kind of decided natural fibers were too limiting. We wanted materials that were completely waterproof, transparent, moldable, so we turned to crude oil to synthesize plastics and synthetic fibers like polyester.
Right, and the problem, of course, is the molecular structure of those oil-based synthetics.
Because oil-based plastics are essentially indestructible on a biological timescale. But now that we are drowning in synthetic plastic pollution, the engineering is pivoting back to plants to create bioplastics, things like cellophane spun from wood pulp or stiff plastics synthesized from the starch of potatoes and maize. But wait, I have to play devil's advocate here.
Go for it.
We realize oil is terrible for the environment, so we're going back to plants to make bioplastics. Is this really the perfect guilt-free solution it's marketed as?
Well, if we connect this to the bigger picture, the reason bioplastics are hailed as the solution is entirely about enzymatic recognition. Synthetic plastics are made of long hydrocarbon chains that do not exist in nature. Soil bacteria and fungi look at a plastic bottle and they don't recognize it as food. They have no enzymes to break those artificial bonds.
Right.
But bioplastics like polylactic acid or starch-based polymers are heavily oxygenated and structurally familiar to them. The decomposers possess the enzymatic keys to unlock them, completely degrading the material back into the ecosystem.
So they naturally dissolve or get eaten, effectively solving the pollution nightmare.
They solve the chemical nightmare, yes, but they introduce a massive societal puzzle regarding land use and scale.
Oh, because of how much we need.
Exactly. To replace the global demand for oil-based plastics with bioplastics, we would need to manufacture them in astronomical quantities. That requires growing immense monocultures of maize, wheat, or sugar beet, not for human consumption, but purely to synthesize packaging.
Ah, that forces a brutal resource calculation. I mean, we have an exploding global population and a finite amount of arable land.
Right.
So do we use our fertile soil to grow food to keep food prices stable? Or do we use it to grow biofuel for transportation? Or, do we dedicate millions of acres to growing bioplastic materials just so our packaging degrades?
And that's the issue. There simply isn't enough agricultural surface area on the planet to do all three simultaneously without causing severe deficits in the others. We report this without taking sides, of course, but it's a stark reality in the source text. The plant's molecular engineering is flawless, but our application of it forces us to prioritize our survival needs.
And our survival doesn't just depend on plant architecture or energy storage. Plants don't just defend themselves with thick wooden walls. Since they can't physically run away from an infection or a hungry herbivore, they are forced to stand their ground and engage in chemical warfare.
Oh, their biochemical defensive arsenals are just staggering. They synthesize complex secondary metabolites purely to deter, poison, or kill threats.
Like cotton plants, they produce a phenol called gossypol that acts as a potent natural antiseptic against microbes. And willow trees flood their bark with salicylic acid to fight off pathogens. Humans figured out centuries ago that chewing on that willow bark relieved physical pain.
Right.
And eventually chemists extracted that defensive compound, tweaked the molecule slightly to make it less irritating to the human stomach, and created acetylsalicylic acid, which we know today as aspirin.
It's amazing. A plant's localized immune response was reverse engineered into a global painkiller taken by millions every single day.
But the most intense example of humans tapping into plant chemical warfare has to be the discovery of digitalis.
Oh, the work of William Withering in 1775. It is a foundational and highly controversial moment in the history of pharmacology.
Yeah, so Withering was a doctor treating a patient suffering from the dropsy, which we now know as severe edema. It's massive fluid retention caused by a failing heart. Withering's treatments just weren't working, and the patient was dying. So in desperation, the patient goes to a local folk healer who administers a secret herbal soup. The patient drinks it and makes a miraculous recovery. The fluid drains, the heart stabilizes.
And Withering, being a scientist, tracked down the healer and acquired the recipe. It contained 20 different botanicals, but Withering's expertise allowed him to isolate the single active ingredient doing the heavy lifting, which was the foxglove plant.
But foxglove is a notoriously toxic woodland plant.
Highly toxic, yes.
It's designed to disrupt the heart rate of animals that try to eat it. So Withering, fully knowing it's poisonous, decides to experiment. Over 10 years, he administers variations of this toxic foxglove soup to 163 of his heart patients. It sounds like a brilliant but completely reckless mad scientist.
He was systematically hunting for the therapeutic window. He documented that if he administered too high a concentration, the defensive toxins triggered severe nausea, vomiting, and visual disturbances. But if he titrated the dose down to a very specific narrow margin, it stimulated the heart muscle to pump more efficiently, clearing the fluid buildup.
So he was walking a microscopic tightrope between a miraculous cure and lethal poisoning. And he isolated the active chemical, digoxin, which is incredibly still prescribed by cardiologists today for heart failure.
Yes, it is.
But I mean, his methodology, handing out toxic soup to hundreds of people through trial and error, you couldn't exactly wander through a hospital handing out toxic soup today to see what happens. It would obviously send the modern scientist straight to prison.
Oh, absolutely. His crude experimentation proved that powerful system-altering medicines exist in plants. But his utter lack of statistical control proved we needed a rigorous framework to validate them safely. Withering's wild trials are exactly why the modern clinical pipeline is so strictly regulated.
So today, a new plant-derived compound has to run a massive, multi-year statistical gauntlet before a doctor can even prescribe it.
It always begins with preclinical testing. Long before human exposure, the chemical is introduced to cultured cells to screen for basal toxicity. If it passes, it moves to animal models. This is a required regulatory step to map systemic absorption.
To see how it moves through the body.
Exactly. We have to see how a complex living mammalian system metabolizes the compound and how the liver and kidneys excrete it.
And if the systemic map looks safe, it enters phase 1 clinical trials. But this phase isn't even about curing the disease, right? It's tested on a very small group of healthy volunteers, purely to monitor for unexpected, dangerous side effects in human biology.
Right. Only after proving human safety does it advance to phase 2, where it is administered to a small cohort of patients actually suffering from the target disease to track basic efficacy. If it shows promise there, it enters the definitive arena, which is phase 3.
Phase 3 involves thousands of patients across multiple demographics. And crucially, this is where the double-blind, placebo-controlled methodology is enforced to counteract human psychology.
Because the placebo effect isn't just someone imagining they feel better, it is a profound neurological mechanism. When a patient believes they are receiving a powerful treatment, the brain's expectation of relief can actually trigger the release of endogenous opioids and neurotransmitters that tangibly reduce pain and alter physiological symptoms.
Right, so if you give a patient a dummy pill with 0 active ingredients, their own brain might start fighting the symptoms anyway. So to prove the plant chemical is actually doing the work, the trial's randomized. Half the patients get the real drug, half get the placebo. But because it's double-blind, the doctors interacting with the patients also have no idea who is getting what.
And that double-blind layer is vital. If a doctor knows they are handing over the real, powerful plant compound, their subconscious body language, their tone of voice, their microscopic expressions convey confidence.
And that rubs off on the patient.
Exactly. That confidence transfers to the patient, artificially spiking the placebo effect. By blinding the doctors, too, the data reflects the true, isolated chemical efficacy of the drug.
It takes upwards of 10 years and billions of dollars to clear that pipeline today. It ensures that when we harvest a plant's chemical weapons, we're actually healing the patient, not just relying on their mind's expectations. So what does this all mean?
Well, we've covered a lot of ground.
We really have. For you listening, we started out looking at that potted fern on your desk, and hopefully you now see the incredible hidden machinery humming away inside it. We went from flipped chemical bonds of beta-glucose to towering woody stems to discovering nickel mining weeds and tracing the path from poisonous flower soups to modern double-blind clinical trials.
It just goes to show that plants are the silent engineers of our world. They are fundamentally shaping our climate, our technological materials, and our medicine cabinets.
They really are. But as we wrap up this deep dive, I want to leave you with one final thought to mull over, something to explore on your own. We spent this whole time talking about how humans actively exploit plants, right? Cutting them down for wood, extracting their chemicals, farming them for bioplastics. We operate under the assumption that we are the master engineers, in total control of the biological world.
Right?
But consider the fact that plants, which literally cannot move, have somehow successfully tricked humanity into obsessively cultivating them. I mean, we clear out their competitors, we invent toxic pesticides to protect them from bugs, we build vast irrigation systems to ensure they never go thirsty, and we transport their seeds across every single continent on the globe to guarantee their species' permanent survival.
It's an interesting perspective.
Next time you take an aspirin or throw a biodegradable cup in the trash or just water that fern on your desk, ask yourself, who is really farming who?